Good day, everyone. Welcome to the section on neurophysiology of the Physiology 1 course at the Atlantic Veterinary College. This section of the course will consist of 6 lectures. We will first look at the role and organization of the nervous system. In the second lecture, we will review muscle receptors and spinal reflexes. Then we will move into the senses of hearing and vision, followed by the vestibular system, the cerebellum, and finally the limbic system. Let's start by looking at the nervous system. Our objectives for today are first to define what the nervous system does and how it is organized. Then learn about the function and the different components of the central nervous system. And finally, address the concepts of synapses and receptors. The nervous system is continuously sensing both the external and the internal environment. The information about the external environment is acquired through auditory, visual, olfactory, taste, and tactile inputs. The internal environment is monitored continuously through many sensors located in different organs and tissues, such as heart rate, respiration rate, distension of the gut, chemical composition of the digestive contents and that of blood, etc., etc. Once all that information reaches the central nervous system, it has to be integrated, and then the central nervous system has to either formulate an appropriate response, may need to commit some of this information to memory, or what is the most common result is to discard the information as irrelevant. The other distinct role of the nervous system is to control the function of both skeletal and smooth muscle, as well as the secretion of different glands in the body. The purpose of doing all what we have mentioned before is to maintain the organism functioning within a range of parameters which is meant to maintain homeostatic conditions, a balance between the environment and the physiological and emotional status of the animal. Finally, the nervous system is in charge of maintaining an animal conscientious, generating memories, and functioning within a given emotional state. Now that we know what the nervous system is supposed to do, let's find out how is this system organized. One component of the nervous system is the central nervous system, and this is made out of the brain and the spinal cord. The brain is located within the skull and protected by the cranium, while the spinal cord is encased and protected by the vertebral column. The other component of the nervous system is the peripheral nervous system. The peripheral nervous system consists of all the nervous tissue, nerves, and ganglions that are located outside the skull and the spinal cord, and it is divided into two divisions. The sensory or afferent division, which, as we mentioned before, brings all information from the environment and the internal organs into the central nervous system. The second division is the motor or efferent division, and this carries information from the central nervous system to muscles, organs, and glands throughout the body. The motor division, however, is divided into the somatic nervous system, which innervates the skeletal muscle and executes voluntary or reflex actions, and the autonomic nervous system, which innervates all the organs and cardiac muscle and smooth muscles of the body to regulate involuntary or automatic functions. The autonomic nervous system is further divided into sympathetic and parasympathetic, as well as the enteric nervous system. The sympathetic nervous system is responsible for making the body react fast under emergency situations. It's associated with the flight and fight reaction. The parasympathetic nervous system is responsible for promoting normal biological activities. Therefore, it is also called the rest and digest system. The last system, the enteric nervous system, controls gastrointestinal activities independent of the central nervous system. The components to trigger a sensory signal can be seen here. For example, in this case, the skin that received the stimulus, a neuron taking the signal to the spinal cord through the dorsal root, and the gray matter where further connections are made. The peripheral nervous system is divided into two large divisions, the sensory or afferent division and the motor or efferent division. The sensory division brings information from the environment and from different organs into the central nervous systems. The neurons involved in the sensory division have their body located in the dorsal root ganglion, and they are either a receptor or connect to a specific receptor. Once the receptor is activated, the information is converted to an action potential and transmitted to the central nervous system. The signal is delivered through the dorsal root to the posterior gray horn of the spinal cord, where it synapses with other neurons to be integrated in higher centers or to elicit a reflex action. The somatic nervous system in turn requires the spinal cord, where the signal is generated, a connecting neuron which carry the signal, and the skeletal muscle to carry out the required action. As we have already mentioned, the motor nervous system carries information from the central nervous system to different parts of the organism. The efferent division is further divided into the somatic nervous system and the autonomic nervous system. The somatic nervous system specifically connects the central nervous system with skeletal muscle. The cell bodies of the neuron carrying the signal are located within the central nervous system, specifically in the spinal cord. Their axons leave the spinal cord through the ventral root of a given spinal nerve and connect directly with the skeletal muscle through a synapse to trigger an action by the muscle. The connections of the autonomic nervous system are a little more complex than those of the somatic nervous system. They involve the spinal cord, two efferent neurons, synapse at a ganglion, and the effector organ. The autonomic nervous system sends information in the form of an action potential between the central nervous system and a smooth muscle, cardiac muscles, and several secretory glands. Two neurons complete the connection between the central nervous system and the effector organ. The first neuron has the body located within the central nervous system and is called the preganglionic neuron. The axon of this neuron leaves the central nervous system through the ventral root of a spinal nerve or from a cranial nerve and synapse with another neuron, the second motor neuron. The body of the second motor neuron, also called the postganglionic neuron, is located in an autonomic ganglion outside the central nervous system, and its axon synapses with the effector organ or muscle. The basic unit of the central nervous systems are the neurons. Here you can see the static image of a genetic neuron with all its components: the dendrites, the cell body, the axon helix, and the axon with its myelinated sheath, and the presynaptic terminals. We will discuss this in a minute. Depending on their roles and their shapes, neurons can be classified as multipolar neurons, bipolar neurons, or monopolar neurons. As mentioned before, neurons are the basic structure of the nervous system. A genetic neuron is characterized for having dendrites, which connect to the body and receive information for that neuron. Within the body is located the nucleus. At the opposite end, there is an axon, which is the route to send information from this neuron to other cells. This information is generated as an action potential in the axon helix, which is placed in the area where the axon leaves the body. Depending on the location of the cell, neurons use different strategies to have their axon insulated in order to speed up the rate of electrical transmission. In neurons of the peripheral nervous system, the axon may contain a myelin sheath, which is made out of several Schwann cells or neurolemnocytes, separated by a space called nodes of Ranvier. In the central nervous system, the axons of neurons are insulated by oligodendrocytes. At the end of the axon may separate into several branches which will connect to different cells. The connections are done through specialized structures called presynaptic action terminals. A neuron with a highly branched dendritic terminal and a single axon is called multipolar neuron. These are usually found in the central nervous system, specifically in the cortex, the cerebellum, the spinal cord, and the autonomic ganglia. They serve either as interneuron or motor neurons. The second type of neurons are bipolar neurons. These are characterized by having two extensions at opposite sides of the body. One end is made out of dendrites terminal, which collect information, and it is guided to the body by a single dendrite, and an axon, which is myelinated and carries signals to other cells, information that is transmitted at the axon terminals. These neurons are a specialized sensory neuron, and as such are found in pathways dealing with vision, gustatory olfaction, auditive, as well as vestibular function. The third type of neurons are called pseudo-unipolar or monopolar neurons. These neurons are characterized by having a single process that leaves the body in branches into two components that act as an axon. The branch reaching the periphery is capable of converting physical stimulus such as sound, temperature, and light into an action potential, which reaches the central nervous system through the dorsal root. Normally, the body of these neurons is located in a ganglion. The entire axon is myelinated. To function properly, the neurons of the central nervous system, as well as those of the peripheral nervous system, require the assistance of a variety of very specific cells, each of which play a very specific role. These supportive cells are generically called glial cells, but depending on their role, they have a more specific name. In the central nervous system, the glial cells are the astrocytes. As you can see in the images, are star-looking cells that provide support to the neurons in the central nervous system and to the endothelial cells which make the physical barrier known as the blood-brain barrier. They also provide a conduit for the transfers of nutrients and oxygen from circulation to the neurons in the central nervous system, and they remove waste product from neurons to circulation. Astrocytes also contribute to removing, through phagocytosis, dead neurons and any debris resulting from a traumatic event. The ependymal cells are secretory ciliated cells which are located in the ventricles of the brain and form the choroid plexus, which secrete and circulate cerebrospinal fluid. The microglia are the smallest supportive cells. They are the equivalent of macrophages within the central nervous system. They phagocytize debris and maintain the health of the central nervous system. As we mentioned while describing the neurons, oligodendrocytes enhance the function of the central nervous system by providing structural support and myelin sheath to axon bundles. A single oligodendrocyte can insulate parts of up to 50 new axons. In the upper picture, you can see a single oligodendrocyte sending their processes to several axons. The wrapping effect can be seen more clearly in the lower picture, where 5 processes are clearly seen wrapping around 4 axons. In the peripheral nervous system, there are other types of supportive or glial cells. The Schwann cells or neurolemmocytes play the same role in the axons of neurons of the peripheral nervous system that the oligodendrocyte carried out in the central nervous system. That is, they provide insulation to the axon of neurons, forming a myelin sheath to speed up transmission of the signals. Schwann cells, however, wrap the entire cell many times around a section of the axon, as you can visualize in the image of a cross-section of an axon wrapped with about 7 layers of a single Schwann cell. The other glial cells of the peripheral nervous system are satellite cells which support, nourish, and protect the cell bodies of the neurons located in all ganglia. In the sensory ganglia located in the dorsal root of the spinal cord, and those of the autonomic system, the sympathetic and parasympathetic ganglia. You can see how they surround each other's cell's body in these microphotographs. Let's now look at the brain and its components. The brain is made up by the telencephalon or cerebrum, which in turn is divided into 2 hemispheres, and these are divided in the anterior and posterior lobes. The diencephalon, which is located ventral to the cerebrum, and it is composed of the epithalamus, the thalamus, and the hypothalamus. Caudal to the diencephalon and ventral to the cerebrum, we find the cerebellum. Then the brain stem, which is made out of the midbrain, the pons, and the medulla, connects the rest of the brain to the spinal cord. The telencephalon or cerebrum is the largest component of the brain. Here we can see the anterior and posterior lobes. The cerebrum is divided into the cortex, which is called the gray matter because it is made up of neuron bodies which are unmyelinated, and the cerebral medulla, which is called the white matter because it is made up of myelinated axons and they appear whitish. The cortex is responsible for starting voluntary motor activity, analyze perception involving the senses, store memories, create awareness, conscientiousness, and thought. The medulla is made up of nervous tract connecting different areas of the brain and the rest of the central nervous system. The diencephalon is located between the brain stem and the cerebrum. It contains the thalamus, which receives all sensory information except olfactory before sending the information to the different parts of the cortex. Thus, it is considered the sensory relay of the brain. Emotions such as rage or fear are processed in the thalamus, thus influencing and integrating the appropriate sensory information and determining mood. The hypothalamus is located under the thalamus, and it contains a variety of nuclei and nervous tracts. Some of these are involved in responding to olfactory stimulus, but the majority are involved in controlling and regulating the endocrine system in conjunction with the hypophysis. Through these nuclei, the animal controls its body temperature, thirst, hunger, sex drive, blood volume, renal function, and many productive functions such as growth and milk production. In summary, the hypothalamus is the main center to maintain homeostasis using the nervous and endocrine system. The epithalamus, which includes the pineal gland, influences several biological rhythms, including those related to reproduction in seasonal breeders. The epithalamus also has the abenular nucleus that participate in innate response to odors. Then we find the cerebellum, which is located in the base of the cerebrum and caudal to the brainstem. The cerebellum is responsible for posture and balance, but the component of balance which involves muscular activity, as balance is monitored and evaluated in the vestibular system located in the ear. The cerebellum also deals with locomotion and fine motor coordination. Complex movements are learned in collaboration with the frontal lobe of the cerebral cortex. Next, we have the brainstem, which links the cerebrum and the cerebellum with the spinal cord. The brainstem is divided into the midbrain, which controls the reflexive movement of the head, body, and eyes in response to sound, texture, or sight. The pons, which serves as a relay of information between the cerebrum and the cerebellum. Furthermore, integrated within this area are the respiratory center and heart rate controls, integration of movement, and many more. The third component of the midbrain is the medulla oblongata, where several nuclei control different functions aspects of respiration, such as the rate and the depth of respiration. It also permits conscious control of skeletal muscles that permit the voluntary inhalation or holding of your breath. In the medulla, many of the nerve fibers cross from one hemisphere of the brain to the opposite side of the peripheral nervous system. The last structure of the brainstem is the reticular formation, which is an overlap cluster of nuclei distributed throughout the brainstem and controls many cyclical activities such as the wake-up-sleep cycles. Changing to something more detailed, let's view how do neurons transmit their signals to the next cell, which could be another neuron or a muscle fiber. This interaction takes place through a connection called synapses. In order to transmit its signal, a neuron has to trigger a depolarization on the next neuron of the path. This is achieved with a synaptic connection. Within the presynaptic neuron, in the terminal button, there are thousands of vesicles containing neurotransmitter. Only a few of them are utilized in each depolarization, so a neuron can repeatedly transmit signals to the next neuron. The type of neurotransmitter in these vesicles depend on the type of neuron. Therefore, there are many neurotransmitters and each acts in a slightly different manner. Some are inhibitory, thus preventing the polarization of the next cell, while others are stimulatory, thus triggering the polarization of the next cell. An example of the general pattern of synaptic activity is as follows. After the action potential is generated in the axon helix of the presynaptic neuron, the action potential travels through the axons towards the terminal button. Upon reaching the terminal button, the action potential triggers the opening of voltage-gated calcium channels located in the membrane of the terminal button. This allowed the rapid entrance of calcium from the synaptic cleft into the terminal button. An elevation of calcium within the presynaptic button triggers some of the neurotransmitter carrying vesicles to empty their contents into the synaptic cleft. The neurotransmitter binds to receptors located in the dendrite or cell bodies of the postsynaptic neuron. The result of this binding is the opening of sodium channels, thus permitting the elevation of intracellular sodium and, as a consequence, a change in polarity of the postsynaptic membrane. The change in polarity within the membrane of the postsynaptic neuron triggers an action potential within the postsynaptic neuron, which moves through the dendrite towards the cell body. After the action potential is started, the neurotransmitter is released from the receptor in the postsynaptic neuron, and they can be either reuptake by the presynaptic button or enzymatically degraded within the synaptic cleft. The last topic that we will discuss in this session has to do with receptors. In the context of sensory activity, a receptor is a cell which can perceive a given stimulus and convert that information into an action potential, which is then interpreted by the central nervous system. From the environment, receptors allow animals to perceive light for vision, temperature, sound, smell, and taste. From within the organism, receptors can detect concentrations of solids, pressure, distensions of tissue, frequency of many events such as heart rate or respiration, and more complex situations such as balance or orientation. Receptors can be classified based on type or the sensation they detect. This slide will be quite busy. I do not expect you, however, to memorize this information. What I would like you to do is to understand the concept of how different sensations are perceived in order to be converted into an action potential, which the brain will eventually interpret. Most common types of receptors are mechanoreceptors. They detect sensation of touch, pressure, vibration, and what triggers this sensation is some type of compression. Here you can see a list of some of the mechanoreceptors. Each responds to a slightly different stimulus. Meissner corpuscles are located very superficially in the skin and respond to a slight touch. Hair cells are able to respond to a stroking of the hair. Pacinian corpuscles are located a bit deeper and they are able to detect vibration, while the Merkel disc or Merkel complex are located slightly up in the skin and they detect pressure and texture. Ruffini sense the stretching or tension in the surface of the skin. We have the organ of Corti, which responds to vibration, and this hair cell type of receptor is located in the ear. The vibration is generated by sound. Many free nerve endings are capable of detecting multiple inputs when you feel, and each nerve ending are perceiving it. And some of those are associated with some type of mild irritation. When the irritation is more significant, then the sensation becomes pain, and it is detected by free nerve endings, which are named nociceptors. In the case of temperature, this sensation is also detected by specialized nerve endings. They are specialized because some nerves detect cold temperature and others detect warm and hot respectively. The combination of all of these inputs are integrated in the brain to determine the actual temperature. Then we have very specialized receptors which are capable of differentiating molecules. These are used to identify smell and tastes, and they are based on binding of molecule to a receptor. In order to see, animals have photoreceptors, which are able to convert different frequencies of light waves into a recognizable signal that can be interpreted as colors and shape by the higher centers in the brain. For the central nervous system to be capable of differentiating the strength or intensity of a given stimulus, it relies on what is called the frequency code and population code. Frequency code refers to the number of events sensed or detected per unit of time. For example, if the pressure is sensed 3 times in 1 second, it has a much lower frequency code than pressure that is sensed 25 times in 1 second. Therefore, the intensity perceived for the second case is much higher. The units used for frequency is the hertz, which corresponds to 1 cycle per second. Population codes refer to the density of receptors in a given area or volume. If you press an area that has 20 receptors per square centimeters, it will be perceived as a stronger signal than if you press an area that only has 4 receptors per square centimeter. Finally, the sensitivity of an area or the ability to discriminate inputs is determined by the population code times the area which a receptor covers. Based on this principle, you will find out, for example, that if you touch your lips with 2 points of a compass separated only 2 millimeters, you will feel 2 points of contact, while if you touch your back with the 2 points of the same compass but separated as much as 1 centimeter, you may feel only 1 point of contact. This is because the lips have a much higher population code that is more receptors per area, and each one of these covers a very small area, while in the back there are very few receptors, but each one covers a large superficial surface area. This brings us to the end of this session. I hope you have enjoyed it. Remember that I am available to answer questions in Blackboard Collaborate during the scheduled hours of class, as well as by email at any given time. In summary, today we cover how the nervous system processes information, whether this is external or internal. We looked at the component of the nervous system and determined that sometimes the organism performs activities automatically, while in other cases these activities are done on purpose. Specifically, we look at the cells involved in the nervous system. Then we reviewed the different structures of the brain and mentioned what each component does. We examine in a more detailed manner what is involved in a synaptic transmission. Then we describe the types of receptors, and finally we explain why we can differentiate the strengths of a stimulus as well as the sensitivity of a given area. Have a good day.